There is a habit of thought in molecular neuroscience that mistakes the endpoints of a biological pathway for its actual mechanism. We measure the chemical inputs, we measure the final outputs, and we treat the physical space between them as irrelevant plumbing. In the study of Alzheimer's disease, this habit leads directly to an analytical dead end. We traditionally measure the Reelin protein to gauge the brain's protective capacity, and we measure the tau protein to track the ruin of the internal cellular structure. This creates a glaring contradiction in the biomarker data. The diseased cortex doesn't lack Reelin, it actually contains more of it than a healthy brain, yet tau still aggregates into destructive neurofibrillary tangles. Resolving this paradox requires abandoning the endpoints. The decisive biology doesn't live in the raw inventory of these proteins, but in the exact physical event required to connect them. The break on tau is a highly specific physical event occurring on a patch of dendritic membrane just a few nanometers wide. 3 distinct molecules from 3 different chemical kingdoms must form a stable, localized grip to transmit a protective kinetic signal across the lipid bilayer. This event requires 3 partners: a central fragment of the Reelin protein, the ApoE2 transmembrane receptor, and a dense sulfated sugar matrix called heparan sulfate. The failure of any one of these partners collapses the entire system. If they cannot hold one another in a precise, multivalent grip, the inward cascade of phosphorylations never begins, and the kinase that mutates tau operates completely unchecked. The pathology of Alzheimer's disease can proceed with Reelin in absolute abundance, provided this single physical connection is broken. The disease is the failure of this microscopic handshake. To understand exactly how that collapse happens, we have to map the healthy interface piece by piece, beginning with the ligand. The active signaling molecule is not the massive 3,000 residue Reelin protein. In the living brain, proteases cleave Reelin to generate a much smaller, signaling-competent central fragment spanning its 5th and 6th repeats. Structural models of this R56 fragment reveal a highly specific contact surface. The physical center of its grip relies on 2 projecting amino acids, lysine 2360 and lysine 2467. These lysines meet the second partner of the clasp, the ApoE2 receptor on the postsynaptic membrane. Specifically, the action occurs at the very first ligand-binding module on its extracellular stalk, the LA1 module. The LA1 module clamps down on the Reelin fragment using a double lysine grip. This binding chemistry is shared across other endocytic lipoprotein receptors to grip their cargo. This single interface buries only about 350 square angstroms. It is a deliberately modest, calcium-dependent contact. A lone Reelin fragment connecting to a lone receptor is easily broken and nearly inert. Firing a potent inward signal requires avidity. The system relies on many of these modest grips, closing simultaneously across a tightly clustered array of receptors. But Reelin and ApoE2 cannot cluster themselves. Presented as individual pairs, they drift laterally in the fluid lipid bilayer, unable to form the rigid geometry required to multiply their signal. They require a scaffold to concentrate them. This is the historically overlooked third partner, a linear, heavily sulfated polysaccharide called heparan sulfate, displayed in dense perfusion along the neuronal surface. The chain is modified in specific patches. The domains carrying N-sulfate groups act as an obligate co-receptor. They physically catch the drifting Reelin and ApoE2 pairs, locking them close enough together to construct the multivalent signaling array. Without this precise sugar architecture, the receptor cannot dimerize, the geometric problem remains unsolved, and the entire protective signal against tau is voided. When that clustered geometry is achieved, the internal cytoplasmic tails of the ApoE2 receptors are pulled tightly together, bringing along their docked adapter molecules, disabled 1. Clustered disabled 1 is then phosphorylated, triggering a self-amplifying kinetic cascade that descends through the SRC family kinases and terminates in the inhibition of the enzyme GSK3-beta. GSK3-beta is the principal kinase that hyperphosphorylates tau. Keeping this enzyme suppressed keeps tau in its normal, healthy state. This specific downward chain is the literal mechanism of the tau break. But the matrix that scaffolds this break contains a dark biological contradiction. The heparan sulfate polymer that stages Reelin's protective signal is the exact same surface molecule that pathological tau aggregates use to enter the cell and spread from neuron to neuron. These opposite functions exist on the same polymer because of the sulfation code. The sugar chain is not uniform. It is a mosaic of differently modified domains written in a chemical alphabet by local enzymes. Reelin reads one specific passage of this text. The protective receptor cluster relies entirely on the N-sulfated domains of the chain. Tau reads a different passage. Its entry into the neuron specifically requires 6-O-sulfation, while the toxic internal tangles it forms are chaperoned by rare 3-O-sulfated domains. One physical polymer bears both the shield and the gate, inscribed side by side in distinct chemical ink. Alzheimer's disease is legible as the simultaneous failure of this clasp on all three of its sides. On the ligand side, rising extracellular amyloid plaques physically trap the active Reelin fragments, sequestering them away from the postsynaptic membrane. This Reelin resistance explains the paradox of the biomarker data. The neuron produces more Reelin in response to stress, raising the total tissue inventory, but the signal remains deafened because the trapped fragments can never reach the receptor. On the receptor side, apolipoprotein E, or ApoE, acts as a direct competitor. It grips the exact same LA1 module on the ApoER2 receptor, utilizing the identical double lysine chemistry. The highest genetic risk factor for the disease, the ApoE4 variant, weaponizes this competition. ApoE4 degrades membrane recycling, dragging the ApoER2 receptors down into intracellular endosomes and removing them from the surface entirely. The most insidious failure occurs on the sugar side. In the diseased hippocampus, specific modifying enzymes, notably HS3-ST2, are aggressively upregulated, causing a drift in the sulfation code. These enzymes rewrite the sugar bed. They erase the intact N-sulfated patches Reelin needs, actively replacing them with the 3-O and 6-O sulfates that serve tau. The multivalent geometry dissolves, the Reelin cluster breaks apart, the protective inward signal dies, and the recoded sugar matrix easily admits the pathological tau seed. A trapped ligand, a sequestered receptor, and a re-inked sugar matrix operate as a single feed-forward collapse. A weakened signal permits more tau, which drives inflammation, which further remodels the sugar. The decisive proof of this three-body model exists in human genetics. The Antioquia kindred in Colombia carries a fully penetrant mutation, causing deterministic, early-onset dementia. Yet two specific members of this family held off dementia for roughly 3 decades past their expected onset, presenting spared cognition despite carrying incredibly heavy amyloid burdens. Their protection stems from 2 distinct secondary variants, the ApoE3 Christchurch mutation and a gain-of-function Reelin variant called Colbos. At the dendritic membrane, both variants execute mirror adjustments to the sugar-facing side of the clasp. The Christchurch variant weakens ApoE's harmful grip on the sugar bed, while the Colbos variant tightens Reelin's protective grip, ensuring the clasp stays securely closed. Because heparan sulfate is the portal for tau spread, developers are pursuing blunt therapeutic agents to block this sugar entirely. But dismantling the matrix to shut the gate will inadvertently silence the Reelin brake as well. The cortex was built on this specific molecular handshake. The therapeutic objective is not to break the matrix, but to read its sulfation code, preserving the domains Reelin requires, denying tau the architecture it exploits. The clasp must be tuned before the structural integrity of the circuit finally gives way.